Hey, good morning. So, couple things. So, we're going to continue talking today about enzyme function, and the book does not go into very much depth about how enzymes actually work. And I like to cover this in a little more detail than you're going to find in the book. So that's what we're going to talk about today. Couple quick things. So, I posted two sets of problems and study questions on BSpace. So hopefully you had a chance to download those. A number of students are asking where are the answers, and I've asked the GSIs to put together the answer keys for those, so those should be posted later this week. I'll check with them today about when those will be up. Look for that in the next few days. I'm also going to be posting another set of problems probably late this week, maybe over the weekend. That'll cover the more recent material that we're covering. Okay, so keep an eye on BSpace for those problems, the next problem set. The other thing is I just want to clean up something from last time. So we had an iClicker question that was maybe a little ambiguous because we didn't actually specify the standard conditions for the reaction. So I'm actually going to give credit for D or E because some people got confused about the conditions for the reaction. Not the standard conditions were not specified in that way that I originally wrote the problem. So if you voted for E, you're still going to get your point. Okay? And there's a detailed explanation of this on BSpace if anybody wants to look at that. All right, so from last time, you remember that we were talking about the relationship of free energy to the spontaneity of a reaction. And basically, if we have a spontaneous reaction, this occurs without energy input. And you know from this relationship that means that the change in free energy of that reaction is negative. So for a process to occur without an input of energy, it has to either increase entropy or disorder, and that's this term right here, or it has to change the total energy, decrease the total energy, which is the enthalpy term right here. Okay, so important to keep in mind this relationship as we think about enzyme catalyzed reactions, because as we'll see, many of these reactions that are catalyzed are not spontaneous reactions. They're actually reactions that have a positive change in free energy associated with them. And we're going to talk about one of the things we'll discuss today is how does the cell deal with that? How do we drive reactions that are actually require energy to occur? And again, from last time, we looked at this reaction coordinate diagram where we have free energy plotted over here on the y-axis. We have reaction coordinate here. We start off with substrates in the ground state, and then they end up as products over here. In this example, this would be a favorable reaction, right? A spontaneous reaction, because the change in free energy going from substrate to product is negative. So we have negative change in free energy. But to get there, there's actually an energetic barrier. And this is called the transition state of the reaction. And what enzymes do effectively is they lower this energetic barrier, the energy of the transition state, so that it makes it easier to get from substrates to products. And they do this by interacting with substrates. And we see a sort of local energy minimum here that represents the favorable interaction of the enzyme with its substrate. And then once it crosses this barrier, this is where chemistry is actually happening here. Now we have products bound to the enzyme, and then they eventually get released during the reaction. And so what we're going to see is that binding energy is actually a major source of the free energy that's used by enzymes to lower the activation energy of a reaction. In other words, there's favorable chemistry between an enzyme and its substrate, and that favorable interaction is one of the things that helps to lower the barrier to reaction occurring. So just a couple more terms. So you know that a catalyst is a chemical agent that enhances the rate of a reaction without being consumed by the reaction, and an enzyme is just a catalytic protein. And I keep harping on this, but it's because it's what I worked on in my past life is catalytic RNA molecules. We actually call those ribozymes, right? So we know that some biological catalysts are actually RNA ribozymes. And then the book shows you just one sort of simple example of an enzyme sucrase that is an enzyme catalyzed reaction that basically is the hydrolysis of a disaccharide, shown here, sucrose, table sugar, to its component monosaccharides down here. And this is a very slow reaction normally, but when we have an enzyme present, it speeds up this rate tremendously. And again, what's very important to remember is that enzymes are incredibly selective. So this enzyme sucrase is specific for sucrose, and it doesn't work on other kinds of disaccharides, even though you might say, well, they're still sugars, they look kind of similar, yes, but they're not exactly the same in terms of 3D structure, and that's really what the enzyme active site is looking at. So we talked about the activation barrier. So you know that every chemical reaction between molecules involves bond breaking and bond forming. And the initial energy that's required to start a chemical reaction is called the free energy of activation, and represented like this. And this is often supplied in the form of heat from the surroundings. Now that doesn't work for biological systems typically, right? Because the cell sort of has probably a uniform temperature mostly across the cell. We probably don't want to boil the contents of the cell to try to enhance the rates of reactions, because that will cause other problems. Protein denaturation, for example, disruption of the plasma membrane. So this is not going to be an effective way for biological systems to catalyze reactions, and that's why we need to have enzymes to do it. And this is just, again, showing another depiction of this energy of activation, in which we have reactants starting off at some value of free energy. They have to go through this energetic barrier to get over here to products. So how do enzymes do this? And this is what I want to spend a little bit of time talking about, is how enzymes actually lower this energetic barrier. And they basically, and again, we said this last time, but it's really important to really get this, that they don't affect the change in free energy of the reaction. They actually just increase the reaction rate. Right? So they increase the rate, but they don't alter the overall change in free energy that occurs in going from substrates to products. And again, this is a slide taken from a figure in the book, in which you can see that we start off here with some value of free energy for the reactants, and whether the reaction occurs along this uncatalyzed path, which is shown in the black line, or whether it occurs along this catalyzed path, which is shown by the red line, the free energies of the reactants and products actually don't change. Okay? And so the overall change in free energy is not changed, but the energy of activation is lowered in the presence of the enzyme. And so the question is, so of course, people have been very intrigued by this for a long time, is how is it that enzymes are able to do this? How can they be so selective for particular kinds of substrates? And furthermore, how do they enhance rates of reactions? And I showed you the table last time that shows that many enzymes enhance the rates of the reactions that they catalyze by many orders of magnitude. Right? So they're really incredibly efficient and effective catalysts. And so one of the things that is really very critical for this kind of activation and rate enhancement is that enzymes are very, very selective for the substrates that they interact with. And so we talked about the fact that enzymes form an enzyme-substrate complex, and when they do that, they position the substrate. And by the way, the substrate could be one molecule, or we could have a bimolecular reaction. We're having multiple, two or sometimes even more substrates coming together in what's called an active site. And this is the region in the enzyme or on the enzyme where these substrate molecules are actually binding. And so we know a lot about enzymes and how they interact with substrates based on X-ray crystallography, where it's been possible to solve crystal structures and get real molecular models of enzymes, both in their what's called their apo state, they're not bound to any substrate, and then in their actually active conformation, where they're bound to a substrate. And very typically, what we see when we look at those kinds of comparative structures is that enzymes will change conformation, sometimes quite dramatically, upon binding to a substrate. And this is a process that's called induced fit. And what's really cool is that, and I try to highlight some of the research that's been done here at UC Berkeley, because I think I want you to appreciate what a wonderful university you're at and how much incredibly important scientific research is going on here, So back in the late 50s, Dan Koshland, who was for many years a professor here at MCB, at that time was doing research at Brookhaven National Laboratory. And he was one of the early scientists who was studying the structures of enzymes and trying to understand, using enzyme kinetics, how enzymes actually work. And he proposed the induced fit model of enzymes long before there were certainly many crystal structures available to look at for these kinds of molecules. So he had to sort of deduce this based on biochemical data. And the idea was that when an enzyme interacts with a substrate, initially there's really a very weak interaction, but there's enough of an energetic, favorable energetic contact that the substrate sticks around and actually induces a change in the enzyme that now creates a more favorable interaction. And that's sort of represented, if we go back to here. So that's sort of represented here, where you have this initially very weak interaction that occurs between the enzyme and its substrate, and then there's an induced fit, a change in the conformation of the enzyme that leads to formation of this complex here. And so what Koshland proposed was that this was really one of the major ways that enzymes are able to lower the activation energy of a reaction is by interacting with a substrate through induced fit. And this is a slide, again, an example that your book shows, where we have two crystal structures of the same enzyme, one in the apo state, so there's no substrate bound in this case, and then one of the enzyme-substrate complex over here. And in this example, what you can see is that there's a very obvious cleft in the enzyme structure that has a shape that fits very nicely with the substrate. Initially, maybe this interaction might not be so tight because this is the structure of this active site is fairly open. So a substrate molecule can enter the active site, but it can also diffuse away. So there's some equilibrium of that interaction. But when it does occur, some of the time at least, the enzyme will actually change conformation and close down over the substrate molecule. So we now have a very well-formed pocket that has many more contacts with the substrate. And these contacts typically would be things like van der Waals interactions, hydrogen bonding interactions, sometimes even hydrophobic interactions that can occur in the active site that create a lot of selectivity for the type of substrate that can get in there and interact favorably. Right? And so this is very typical for what we see for enzymes. Is that in cases where we actually are fortunate enough to have these kinds of high-resolution structures of sort of a before and after snapshot of substrate binding, we see evidence for this kind of conformational change. So certainly Dan Koshland was very perceptive in proposing this model long before we had actual structures to look at. And this is certainly one of the very important ways that enzymes are able to function through induced fit. Question? Right, so he's asking, do substrates just enter the enzyme by chance, or is there something else that's going on? And the answer, I guess the short answer is we think it's mostly just by diffusion. Okay, so it's just diffusion limited. Now sometimes that's not the case, so sometimes there are accessory proteins that might help load substrates into active sites, and there's certainly many examples of that. But in the simplest case, we just think that, and certainly when we're doing biochemical experiments in the laboratory, we're just looking at a situation where we're just diffusion limited. Yeah. Say again? Diffusion by the substrate, that's right. It's really the formation of that enzyme substrate complex. Yeah. So this is just kind of a summary of some of the different ways, sort of once we actually get this induced fit, we've got the substrate in there, then what happens? What is this thing actually doing? What's going on in the active site? Sort of like magic, right? But it's actually a combination of things. And what's been found through studies over the years of many different substrate enzyme complexes and looking at the kinetics of these reactions and the structures of these enzymes in different states of reaction is that there really are several important ways that the reaction can be enhanced, the reaction rate. One is orienting substrates correctly for reaction. So rather than, let's say we have a bimolecular reaction, rather than two substrates having to diffuse together and only every now and then are they actually aligned correctly for chemistry to occur, what an enzyme does is actually combine to these substrates such that they're actually oriented properly for chemistry. So that's one of the very important ways that enzymes contribute to the rates, speeding up the rates of reactions is by orienting substrates in the active site. Another way that they enhance reaction rates is by creating strain in substrate bonds. So again, when they bind, and especially if there's an induced fit going on, there are a lot of favorable interactions that are occurring between the substrate molecule and the enzyme. But sometimes those favorable interactions are actually sufficient to create some strain in the substrate. In other words, they affect the ground state of the substrate such that it might be pushed into a structure that's closer to what happens in the transition state. So by doing that, they actually favor the structure in the transition state and thereby lower the activation energy to get there. Another way is that they provide a favorable microenvironment, and really this just means that they create a little vessel or enclosure where chemistry can occur. So rather, again, sort of thinking about the diffusion idea, rather than substrates having to randomly bump into each other, they're actually concentrated together in the active site through these favorable interactions with the enzyme. And so they're concentrated there, and they're more likely to interact just because they're right there, they're localized in the same place. And then finally, sometimes there's actually covalent bonding that occurs between the enzyme and the substrate. There's lots of really nice examples of this where there's a transient covalent bond that occurs that forms between the enzyme and the substrate that, again, helps to do some of these various things, provide binding energy, provide orientation of the substrate, help concentrate multiple substrates in the active site. And then very typically that bond is transient. It will break again during the reaction so that the enzyme is restored to its starting state at the end of the reaction. It's ready to catalyze the next substrate or pair of substrates. So I thought I would show you just one little movie. And this is a very sort of classical enzyme. This is an enzyme that people in my lab hate because it's an RNase. It's a ribonuclease. Okay, so its job is to break phosphodiester bonds in RNA. We're a lab that works on RNA molecules, so we don't like RNase very much. But it's a very cool enzyme, actually. And the way it works is it actually has two histidine residues, two histidine amino acids that are in the active site of the enzyme. You have to kind of picture this as a three-dimensional structure, and you've got these two histidines. And as we run this little movie, it's going to show what we think happens in terms of the chemistry during a catalytic reaction. Okay, so we have the substrate coming in. Here's the phosphodiester bond. So it's coming in. We have one histidine abstracting a proton here, and that activates this as a nucleophile. So it can attack the adjacent phosphate, and this histidine then donates a proton to the leaving group. This leaves. We get a cyclic phosphate product, a 2' 3' cyclic phosphate product. And then in the second step, a water molecule enters the active site, donates a proton back to this histidine, and in the process, we actually then get this kind of reverse reaction that restores this histidine to its starting state and opens that cyclic phosphate. And so we end up with a cleaved RNA cleaved bond there that leaves a 3' phosphate group on the RNA. And so there's lots of experiments that have gone into trying to dissect a reaction mechanism like that. It's really cool when you see it kind of drawn out this way. But to actually figure that out, there's lots of experimental data that go into that. So people have done things like actually labeling those histidines so that we can follow where those protons are actually going during the reaction. And that's how we know. That's how we can sort of follow who's donating a proton and when in the active site. And also there's lots of experiments that have been done using substrate analogs, so things that look like an RNA phosphodiester bond but can't be cleaved, for example. So you can actually trap those kinds of substrate mimics in the active site and then look at a crystal structure and try to infer from that how things are actually oriented when you have a true substrate in the active site. So those sorts of experiments would be critical for figuring out these sorts of enzyme mechanisms. So this is a cartoon that is actually taken out of the Lehninger biochemistry book. It's not a perfect drawing because in terms of the way that it tries to depict what's happening in an enzyme active site, but it does convey this idea that at least in some cases, we think that enzymes have really evolved to bind better to transition states than to substrates. And so again, if we look at sort of a diagram here, very schematic picture of a reaction, and they're showing you a substrate drawn as a metal stick that's being broken. It's going through a bent stick transition state. And we have some free energy diagram over here that we could draw. It looks like this. If we had an enzyme that was perfectly complementary to the substrate, in other words, it formed a very, very stable, energetically stable interaction with the substrate, then we might have a situation that we could represent over here on this diagram that would look like this. So we have a very favorable interaction between the enzyme and the substrate, and it's actually so favorable that nothing ever happens ever again, right? So basically they get together and then it's done. Nothing happens. And that actually would obviously not be a good enzyme, not even an enzyme at all, if it got trapped that way. And so really the job of enzymes is to have enough affinity for the substrate that they actually can get together in the first place, and that's this idea of a deuce fit. So there's some favorable energetics there, but what really happens once this enzyme-substrate complex forms is that there are additional interactions that actually favor or sort of you could say strain, I guess, in this example, the structure of the substrate so that it's closer to the structure that occurs in the transition state. So it actually lowers the activation barrier by providing binding energy that favors this the structure in the transition state. And so if you represent that on an energy diagram, we can draw it like this, where we have a favorable interaction, but it's not so favorable that we can never get out of that energy minimum there. And instead, we actually have a favorable interaction with the structure of the transition state such that this energetic barrier is lowered. So this would actually be a pretty good enzyme because it would help this complex to traverse this path and get over here to products. And so, so one of the things that's been kind of cool over the years, so back when I was in graduate school in the 1980s, there was a lot of excitement about this sort of idea in enzymology, how enzymes work, and people thinking about these sorts of models for enzymes. And one of the ideas that came out of this was, well, if this was really true, then it should be possible to find molecules that were evolved to bind very, very well, very tightly to transition states, and those might actually be good enzymes. And one of the natural types of molecules that evolved in biological systems are antibodies. And so one of the ideas that was very exciting at the time was, could we use antibodies to evolve proteins, antibodies that would be complementary, have shapes that would recognize transition state analogs, or let's just start with transition states of reaction, and then they would then maybe be really cool, good enzymes. And so that could be a really great way to make new enzymes if we could just identify transition state structures and evolve enzymes to recognize them. And of course, transition states, we've already talked about this, they're typically very transient, right? They don't stick around for very long, they're very unstable. And so what we have to do instead is we have to figure out ways to design transition state analogs, chemicals that are stable, but they look like the structure in the transition state. And I saw a hand up, yeah. So he's asking, can enzymes also be used to slow down or halt or alter reactions? Well, certainly enzyme inhibitors will do that, and we'll talk about that a bit on Friday. And there certainly are binding proteins that interact with enzymes and will affect their ability to catalyze reactions. So I would say that it's more has more to do with the kinds of molecules that interact with enzymes rather than enzymes themselves. Typically when we talk about an enzyme, we're talking about a protein that has evolved to be a really good catalyst. So this is just a slide I wanted to. These are not in the notes, by the way, but this is just for fun. Okay, this is fun. So this is just showing you a reaction path with a proposed structure of the transition state molecule here. Again, this would be very unstable and not have a very long lifetime. So what one can do is to synthesize a analog of this transition state down here, which is a stable molecule. This is work done by the Hilbert lab, by the way, in Switzerland. Very good chemistry lab. And so they have made these sorts of transition state analogs. And of course, many others have done this too. Peter Schultz, who used to be here at Berkeley and is now at Scripps, did a lot of this work back when he was here in chemistry at Berkeley. And then what you do is, then how do you use this to generate an antibody? And so then what you can do is you can actually take these kinds of analogs, and you can see in this example, this is a small molecule. So it turns out that the immune system doesn't really recognize a small molecule like that very well. So what you have to do is couple it to a larger molecule, we call it a hapten, that allows you to then immunize an animal like a mouse. And so we immunize the mouse, and then we can, through a process that is diagrammed very crudely here, we can actually identify and isolate antibodies that are made in the mouse's body and use them to produce ultimately monoclonal antibodies. That means antibodies that are clonal. They all look like, each molecule of this antibody population looks like every other. Okay, so they're all the same. And they've been evolved to have very high binding affinity to this transition state analog. And then the experiment is, after you do that, to then take those monoclonal antibodies